Moscow team. I'm a team leader. My name is Dasha. Most of our team is from Moscow State University, named Lomonosov. But we have also students from some medical universities and some people from school. Because there are not many teams in Russia, so we decided to find as much people from other universities as we can. So, if you know, there are only 2 teams from Russia this year, so we were the first one. And our project is about detection RNA viruses. So we hypothesized that if we'll detect some special strains of viruses, it will benefit the treatment. So we found out that we started with SARS-CoV-2 virus because it's all around the world and it's a really important problem. And then we made some research and found out that yes, there is such a problem with SARS-CoV-2 virus. There are several strains, there are lots of strains, and some of them are more severe than others. But then we found out that there's also Hepatitis C virus, and there is a special strain in Russia. So we decided to make research about this strain. It's named, it's a recombinant strain, RF2K1B. And the determination of this strain can benefit the treatment. So we decided to make a detector for this virus. And a few words about a detector itself. Oh, I'm in horizontal formats. Oh, I'm sorry. So, it will be such
much better.
I'm sorry. Yeah, I missed it really. Sorry for that.
No problem.
Okay, so so we decided to make a detector based on a Cas system. So we can take a D-Cas protein, Cas, which is broken, and this Cas protein, with help of guide RNA, will find our sequence if it's present in the sample, and we'll see some color, and we'll find out do we have such a problem with this patient or not. Yeah, so it's in short, it's about our project.
Yeah, I think it's a very cool idea. I think the world definitely could use something like your device. You mentioned that you were using CRISPR and Cas9. So what is your methodology involving CRISPR, and what organism, if any, are you using right now for your research?
So now we decided to go deep in bioinformatics, and we started we started with our a sequence that will determine in RNA viruses, in SARS-CoV-2 virus, and in Hepatitis C virus. So we started with bioinformatics. It means that now we do not have any model animals or something like that. And about the method itself, so we'll take our Cas9, then it would decas. So 2 decas proteins with help of guide RNA, they will attach to our sequence, and these 2 decase proteins, they will be linked through beta lactamase. So it will be a split beta-lactamase, and it will join together the splits, and we'll find out the enzyme activity, and we'll have a change in the color. So this is a system, and our system is better than PCR or sequencing because it will be. We hope it will be really cheaper, and you will not need any lab equipment or many people to perform PCR or sequencing. So just a small machine and this 1 hour reaction will be inside it.
Yeah, I think that's a great proposition for your methodology, and I like that you're using the bioinformatics, and I like that you guys are considering affordability as well. Would you have to, because you're targeting specific viruses and different regions have different strains of viruses, are you building a sort of strain library?
I don't think about a strain library, but we'll have just some sequences, like sample sequences, not
Well, let
the
me
whole viruses, of course, but parts. So DNA library, yeah, DNA or RNA library. It's better to say.
Okay.
Not.
I was going to say, rephrasing that, it would be, I guess, a compilation rather of all the viruses you're targeting within your device.
You mean in one device, or?
Right. So you're targeting hepatitis C, HIV, SARS-CoV-2. Would this be all in one device or multiple devices?
Yeah. Yeah, got it. Thank you. So we wanted to make really multi-virus device, but not in case different viruses in one device, but different strains of one virus inside one device.
Right,
So,
okay.
yeah, for example, we found out that in Russia there are one major strain, RF2K1B, and there are a couple of minor strains. So we'll make a detector for all these strains, the most popular of them. For example, 3, I guess. Yeah.
It's fantastic. Very, very efficient. Very efficient process for testing. So in the future, what does further development for a product like this look like, maybe in clinical or industrial pharmaceutical development?
It's hard really to say now because we only starting our projects, and through iGEM we will start it and we have more feedback. But I guess that so our aim is to have all our detectors in clinics, and we want to have these devices for home usage. So to anyone can buy it and test the cells. So I know that there are such tests for HIV, and they're rather popular and they're more simple. There's no need to go into the hospital, and we want to make such a system. So there's self-care tests, yeah.
Excellent. I think that's really great, facilitating it for people to have at home, especially with everything going on with SARS-CoV-2. If we could have easier access to testing, that would facilitate a lot of our statistics and health awareness.